Hi all, Stu Adler here with a brief orientation video for the UO2 lab, since we're remote this week. This video highlights some of the key facilities in the lab, as well as covering some important safety issues. I'll also touch on the 4 experiments you're likely to be involved with. Hopefully you find this video useful in writing your planning memos, as well as finding everything when you start experiments next week. The first thing you should do when you enter the lab is head over to the locker room area, which is to the left of the door as you enter. This area provides a place to hang your coat and put your backpack, and a place to suit up with the appropriate personal protective equipment, also known as PPE. As I mentioned previously, lab safety glasses with splash protection and nitrile laboratory safety gloves are required at all times when you're in the lab. You're responsible for getting your own safety glasses, but gloves are provided by the department. To the right of the locker area is a large blue cabinet. This cabinet contains many of the consumable supplies you'll need, including nitrile gloves. To the right, as you enter the room, are a number of important emergency resources. These include a first aid kit, an emergency shower, and our emergency eye wash station. Hopefully you'll never need any of these resources, but this is where to find them if you do. In particular, if you or a colleague get ethanol or other foreign substances in the eyes, the eye wash will help dilute it or sweep it out as quickly as possible. To activate the eye wash, place your eyes over the upward-facing spigots and push the lever, while a colleague or a TA calls 911. In case of fire, you'll find a fire extinguisher immediately to the right of the main sliding door as you face outward. Ideally, you should never have to use any of these emergency resources because your experimental plan and practices are designed to avoid creating any of these types of hazards. Personal protective equipment is one part of this, but another is ensuring safe storage, use, and disposal of laboratory chemicals, including toxic or flammable liquids. To help with this, the UO lab is equipped with a flammables cabinet located back here, near the opposite corner from the main entrance. You can't miss it. It's the yellow metal cabinet labeled flammable in big red letters. Inside you'll find our supply of freshly purchased and denatured ethanol, as well as a sample storage area where you can put labeled vials of ethanol water mixtures you collect during the course of your experiments. Note to self: clear out these unused solvents so the students have somewhere to put their samples. Another important part of safe storage is properly labeling all substances so that you and others know what is in any containers. The label shown here is of questionable utility, since without access to your lab notebook, the label doesn't mean anything to anyone else. Good sample labels should include all molecular components, have a traceable origin, such as your lab group, as well as any references to details in your lab notebook. Useful tip/warning. Ethanol, which is the main ingredient in most hand sanitizers, is very effective for removing Sharpie permanent marker ink from glass vials so that you can reuse the vials. But be careful not to lose your labels inadvertently. Sample vials, as well as racks to store sample vials, can both be found in the big blue cabinet. For mixing and handling larger volumes of ethanol and water, you'll find an array of available glassware hanging over the sink on the wall across from the main entrance. As a flammable substance, ethanol poses a significant potential fire risk at any significant concentration, even when mixed with water. For this reason, ethanol should never just be washed down the drain. On lab days, you'll find labeled storage containers for waste ethanol or ethanol water mixtures near the sink. There's also a sharps waste bin for any broken glassware. The lab has a large trash can for non-hazardous dry waste, such as used lab gloves and dry paper towels. However, do not dispose of any potentially hazardous substances, such as sharps or items soaking with ethanol, in the trash. This is a violation of UW environmental health and safety regulations, and also a fire and injury hazard to students and staff. Now that we've gotten some of the basic UO safety stuff out of the way, let's go over some key lab resources you might find useful in your experiments. On the tables along the wall opposite from the entrance, you'll find most of the analytical equipment. This includes 2 gas chromatographs for accurately measuring the composition of ethanol water mixtures. You can read more about this in the SOP, but basically you vaporize a small sample of your solution and flow the gases through an absorption column. The ethanol and water travel through the column at different rates, allowing them to be separated and quantified using a calibrated detector. The GCs work best at water concentrations less than 50% and is most accurate for quantifying trace water in ethanol. For larger samples with high water concentration, we also have a densitometer, which can be used to quantify the amount of ethanol in water by the absolute density of the solution. There's also a microbalance nearby for weighing out small solution samples or calibration standards. Both the GCs and the densitometer are delicate and expensive analytical equipment. Please read the SOPs for these instruments thoroughly before using them, and when in doubt, ask a TA or instructor for help. If you break something, it could be very expensive and time-consuming to fix, and very disruptive to you and your classmates who rely on these instruments for their data. Okay, so that brings us to a tour of the 4 experiments we have in the UO2 lab, 3 of which you'll be involved with at some level over the course of the quarter. The first is our pilot-scale see-through distillation column, which is a real steam reboiler and actual trays and downcomers similar to those used in a commercial column. What's really great about this column is that it is mostly controlled manually with valves and knobs instead of just being stuff on a computer screen. And because it's made of glass, you can see the gas and liquid in the boiler and on the trays. For convenience, there's a bare-bones data acquisition system for tracking tray temperatures and flow rates. There's a ladder for climbing up and taking gas or liquid samples from the trays once it's reached steady state. But that's about it. I personally think this is one of the most fun and tactile experiments in the lab, where you really get to nerd out as a chemical engineer. Please be sure to be careful as you're climbing up and down the ladder. To the left of the tray column is our batch distillation experiment. Like the tray column, you'll be separating ethanol and water mixtures, but here it's done in small batches under transient operation, boiling mixtures from an electrically heated pot and separating in a column which uses a spinning band to achieve continuous gas-liquid contacting up and down the column. The column can be operated at steady state at total reflux, or some of the distillate can be removed at a user-controlled rate and collected in a receiver for a composition analysis. There is a data acquisition system for tracking temperatures and controlling vapor rate and reflux flow. This experiment is also a lot of fun and really requires you to think carefully about non-steady material balances and equilibrium. Please, please, please be careful as you work, as everything is made of glass and thus presents a Sharp's hazard if it breaks. Over here is our third experiment, a packed column absorber for removing ethanol vapor from air using cold water as the absorbent. The setup is equipped with a large liquid bubbler, which takes in house air and saturates it with ethanol vapor. The gas mixture then flows into the bottom of the packed column and flows upward through the packing to the top, where it's released to the room. Fresh water is sprinkled down from the top and hopefully absorbs most of the ethanol vapor. Otherwise, the room starts to smell like a Scottish distillery. This is a very flexible experiment because the entire column can be pulled out and the packing replaced with whatever you like: metal wool, ratchet rings, saddle rings, lots of different sizes. So a lot more control over the process hardware than the other experiments have. And again, manual control and sampling, with a bare-bones data acquisition system for tracking flow and temperatures with time. Finally, we also have a shell and tube heat exchanger. Some of you may have had some experience already with the Armfield system in 436, where you studied a double pipe heat exchanger. But here the focus is somewhat different, as we're less interested in the detailed physical principles of heat exchange and more on the real-world performance of a heat exchanger as a process unit and what factors this depends on. Of all the experiments, this one is probably the simplest and most straightforward as an apparatus. Just 4 temperatures and 2 flow rates, right? Well, maybe. But is it optimally configured for getting to the information you really want or need to know? The simplicity here may be deceptive. Anyway, I hope you enjoyed the tour, and I look forward to seeing you next week in the lab. Please stay tuned for another companion video on conducting a hazard review and planning experiments. See you next time.